Well, before I dig in, I wanted to give a little bit of background even on the title of this talk itself. I'm a toxicologist, I'm not a nutritionist, and I have been responsible over the last few years for integrating evidence-based practices into our firm. And as part of that, we have been using systematic review more. And I think this is notable, particularly for this project, because relative to, say, medicine and nutrition, systematic review is very new in toxicology. Every day, more than 50 publications come out in PubMed alone on systematic reviews in the other disciplines, but in toxicology, we have yet to achieve even 50 systematic reviews. So it is very novel from that standpoint. There's been a series of efforts on behalf of health entities and academic groups worldwide on trying to adapt existing methodologies to toxicology. This is needed because, similar to the difference between medicine and nutrition, we are fundamentally often asking different questions. We are asking about adversity, we are asking about safe levels. And along with that, our evidence streams are often very different. So while it is uncommon to deal with large data sets of human data, which we are fortunate to have here, we are often dealing with animal data or in vitro data. So there have been efforts on behalf of the National Toxicology Program, the Office of Health Assessment and Translation that Dr. Myers noted, as well as the European Food Safety Authority has also issued guidance. The World Health Organization right now is currently developing guidance on systematic review. And through the practice of conducting this systematic review, as well as a number of others in our firm, I've been involved in a number of these committees providing feedback as we're still gaining experience in this field. With respect to disclosure, I was, of course, a member of the ILSI North America Caffeine Systematic Review team, and ILSI North America did provide support for me to attend this meeting and present. Without going much further, I do want to take a moment to acknowledge the large multidisciplinary team. As Dr. Myers walked us through the roles, I think it's very important to emphasize that everyone played a significant role in this manuscript. And in particular, I wanted to call out Dr. Candace Stopker. This has been her vision for quite some time. She has a long history with food safety and caffeine safety, and she was disappointed that she couldn't be here today. So in this talk, I'm going to focus on essentially the methods section of our manuscript, or the project, and that involved initiating the review, finding and assessing the studies, and then integrating and developing syntheses. And at the end, I'm going to offer some thoughts on some of the challenges that we encountered, as well as the solutions that we found for these challenges along the way. So digging into initiating the review. So as Dr. Myers noted, we followed the IOM standards, and there are several standards, and today, right now, it's a little small, but we're going to focus on, jump right in. Dr. Myers covered essentially many of the conflict of interest, et cetera. We're going to jump in right here on developing the protocol itself, which begins with topic formulation. And I would like to impress upon you the amount of thought that went into this prior to starting. It's very critical to have a well thought out topic. And this was something that involved discussions with ILSI North America, as well as internally, as what the best question could be. What is the best way to go about this? And time after time again, we came back to the utility of the NAROT paper, which Dr. Keefe introduced. It's a very highly cited reference. Its utility is that it looked at across a number of outcomes or endpoints. It looked at a lot of data, and it then relatively simply gave safe levels. And so we wanted to, in the end, we wanted to update that. We wanted to follow that model because that was of greatest utility to the science field, we felt. So we developed our framework, or the structure for our assessment based on NAROT. So essentially we wanted to I'll draw your attention to the middle, the comparator, the exposure levels that we discussed. Those all came directly from NAROT. So NAROT's conclusions were based on safe levels per day for various populations. We looked at 4 populations: adults, healthy pregnant women, healthy adolescents, and healthy children. Now, NAROT did not specifically identify adolescents in their review, but we knew that it was a category of interest to stakeholders and sponsors, and we wanted to include that. We then had to select the outcomes of interest. We selected 5: acute toxicity, bone and calcium, cardiovascular behavior, and reproduction and developmental. And this was based not only on the results of the NAROT paper, but much of the research that has been done over the last 15 years, highlighted also by Dr. Keefe of the areas of interest. And so some of the areas that NAROT covered we felt were of lesser importance, or there had been sufficient research that we could focus on these endpoints. And then our question was to essentially determine if the NAROT conclusions hold true today. We also, consistent with the OHAT framework of conducting systematic reviews, we did want to look at pharmacokinetics and pharmacodynamics, but this was a contextual topic. So while we did identify the literature systematically, we did not review it in the complete systematic mode, but our goal was to try to find data that advanced the understanding of kinetics and dynamics, because the actual kinetics of caffeine are very well understood, but what we wanted to know is, has there been research that would help us to understand or interpret some of the potential effects associated with our outcomes? So shown on this slide is the generic structure of our PICO questions. And so we were asking essentially a lot of questions, but we were asking for populations, and we had 4 again. Asking if the caffeine exposure for the given population, so 400, 300, or 2.5 mg per kg per day, what the data showed for those levels relative to our outcomes. And throughout the day, you'll see this, particularly when we go through after the break, the examples for each outcome, how these were structured. I do want to take just a short moment. Dr. Myers did bring this up, but it can be confusing. The terminology that we're using, the outcome is broad, so for example, cardiovascular, and then the endpoints are more specific. So this just shows the number of the endpoints that we looked at in the final manuscript for cardiovascular, so there were quite a few. Okay, moving into finding individual studies. So as a reminder, this involves conducting a comprehensive search, documenting the search, managing the data collection, screening and selection studies, making sure that all of that is not only developed, we followed an a priority process, but then documented our path along the way. We searched 3 databases, and this was specific to the dates of January 1, 2001 to June 8, 2015. So we picked up essentially where we thought it was reasonable that perhaps NAROT left off. And we searched PubMed, Embase, and the Cochrane database. Of course, the majority of our literature, primary literature, coming from PubMed and Embase. We searched these using both outcome and database-specific syntax. And again, these were developed with an expert, systematic review expert librarian. We thought a lot about how these were actually going to be run, however. This graphic, which I would not expect you to read, but essentially it's just a figure that shows our search syntax. You can see that it's almost 2 pages long, and the colors represent the syntax by outcome. So one of the ways that we went about this is we ran this in a concatenated form, so we essentially looked for all of our literature in all one swoop. And this was more efficient than going one at a time because we would have more than quadrupled the number of hits that we would have had to screen. And then once we had all of our information, we put that into Distiller SR. If you're not familiar with Distiller SR, it's a software program that is designed specifically to help facilitate systematic reviews, and in particular the front end with respect to screening, selection, and data extraction. It's it's somewhat like an Access database in that it has a lot of flexibility, but it has high functionality. It is web-based, and it has an audit log, so all of our changes were tracked. We know who did what, when, and what got changed and accepted, et cetera. And it allows for a fair amount of customization when it comes to what you want to extract and how you want to do it and how you want to report it. I will also note that we explored different software tools, but given the complexity of this assessment, we needed something relatively high-powered. So before we even got into the actual search itself, we had extensive piloting exercises. And so this involved not only discussions with the librarian, but looking at, not only how many hits are we getting, but what are the types of hits are we getting. We looked to find, are we getting key data sets? We were trying to validate our literature search to make sure it made sense. So after a fair amount of piloting, we, of course, conducted the official search. We then removed duplicates from our assessment. We did this actually via EndNote as a nice dedupe function. And then ToxStrategies went ahead and started digging in with the initial screen of the titles and abstracts. This is notable because many systematic reviews of very large evidence bases start with the review of titles only. And as I hope to show you later, you can miss a fair amount if you're only looking at the titles themselves. Once we got through that phase, we had an internal QA process as well as a relatively extensive committee review. We actually met daily to go over any sort of conflicts that we had, and that involved not only evidence analysts within ToxStrategies, but our caffeine expert. And then the findings at this point, based on titles and abstracts, went out to our subject experts, and so they were provided essentially Excel files and different materials to see what we had looked at and what we included and excluded. Then following that, we went ahead and got the full text papers of everything that was still included at that point. We went through the process of making sure they still met inclusion criteria, and if so, moved those forward. Again, having another subject expert review at that point, looking at the full text decisions, and then ultimately had our bucket of included studies. So a little about what this looks like. This is a screening form example from Distiller. It easily pulls up the title and abstract, and this is customizable. You can put any type of information in that you would like. It also brings up the abstract and with key words highlighted, so it was easy to draw the reader to different terms that you can set. And then we had a dynamic form. So we asked about 15 questions about every title and abstract, and that allowed us to be not only efficient to say if it, for example, does or doesn't involve caffeine right away, but then if it does, we tried to ask some more questions to help us categorize and develop the process for the remaining of the review. So as Dr. Myers mentioned, we had study screening, inclusion, and exclusion criteria that were developed a priori and reported out in the protocols. Now it's important to note that we have the 5 different protocols all contain slightly different, they have a key set of information which I have shown on the slide, but each of the outcomes also has some unique criteria to them. So whether you might have needed to make sure that the exposure duration was at least 6 months to look at bone and calcium, or similar such examples. A very primary and important inclusion criteria is that the study itself had to have a quantitative estimate of caffeine exposure. Now this could come from caffeine directly or from a variety of sources of caffeine. It must have reported an effect on the outcome of interest. It had to be a healthy population, which was defined as not having been hospitalized or diagnosed with disease and or receiving medical treatment at the time of the study. There's a fair amount of literature, as you might expect, on caffeine being consumed along with other substances. And so we had a fairly strict criteria, and it involved essentially 2 pages of guidance on our internal notes on how to evaluate the study looked at caffeine alone. So mixtures with, for example, alcohol or other drugs were excluded. I will note that in some cases, if there was a study where they were looking at co-exposures, if there was a caffeine-only exposure, we would include that study and use only the caffeine exposure group from that data set. Studies had to be available in English. Case reports were included only for acute data. This is somewhat of a non-traditional exception, but it is well documented in the literature that acute data, or sort of case studies or case reports, are important for rather rare events such as death. And our other key criteria is that you have to present original data. So we did not include reviews, of course, in the review itself. We kept some for context. We did, however, include meta-analyses that provided original data. So with respect to getting through the full text papers, once we had the papers in hand, we again went through and made sure that they still complied with inclusion and exclusion criteria. We then moved into generating our evidence tables or conducting our data extraction. So as Dr. Myers presented, we had two sections of our forms. Essentially, the first was very basic information, directly reported by the author, the objective, the methods, the results, the statistical methods, etc. Then we had a second section of our form that was more unique to informing our PICO. So this was our customized information, which may have required some interpretation. So we took a fair amount of notes on this. The majority of this information was aimed at what information are we going to pull from this study to compare to NARAD. Essentially, what is the one thing this is saying about the NARAD conclusion? Is it supporting it? Is it not? Of course, it's never that easy to just have black and white answers because there's lots of information presented in every study. So we were really challenged with pulling out the most refined set of assessment or the most refined conclusion relative to our question. And so this was the section of data extraction which we recorded that information. The other thing that I will call out here is that we wanted to do some we wanted to show some quantitative comparisons. While we weren't doing a meta-analysis, we wanted to be able to show, qualitatively, what was the study showing was, for example, a safe level or not a safe level, and how, so we can compare that to NARAD. So we selected these no observed effect levels or lowest observed effect levels, no L's and low L's. I will point out that all of the data that we extracted is publicly available on the AHRQ's SRDR. Just a small comment here on the standardization of caffeine content. So within our body of evidence, there were quite a few study types and ways to look at caffeine. Our objective was to use the data as reported by the authors. And specifically, if the authors either did an analysis based on caffeine content or they somehow converted their data to caffeine content, we used that. So for example, if the analyses were done on number of cups per day, but then they gave in the paper how much they thought was in a cup of coffee, we used that information. However, if there was only, again, for example, the number of cups of coffee per day, we standardized those metrics. And so we had, and this is in the supplemental materials of the manuscript, we had a decision tree as to when we would standardize and then what the levels that we would standardize were, not only for the caffeine content, but the volume or mass of the substance. Okay, so moving into assessing the individual studies, this focuses on this last part of IOM Standard 3, the critically appraising each study, which involved looking at risk of bias, but also the relevance of the study's population, the relevance to your question. We did this in three ways. We looked first at risk of bias, or internal validity, using the OHAT risk of bias tool. Again, this was released right after we started the project, which was timely. And the reason that this was selected, this tool itself was selected, is because it's designed for our types of research questions versus there are many risk of bias tools out there and available. But again, this was more unique to toxicology. The other advantage to this tool is that it provides guidance on how to answered the questions, which allowed us to have more uniformity in making the risk of bias assignments. We then, for each study, looked at the external validity, tried to provide a characterization of how generalizable it was, how direct was it, how well was it designed to answer our question. And this is something that's from grade and translated into the OHAT handbook as well. Much of the OHAT handbook is based on grade. And then the level of adversity also became very important. And this was a categorization of, for example, physiological or clinical effects, and a categorization of how important is this in decision making. And so this assisted eventually in helping us weighting the endpoints. The example that grade gives is something of lesser importance might be flatulence, and then of greater importance or high importance would be myocardial infarction. So we followed that system as we went through the papers. So the risk of bias tool, again, is set out for both human and animal studies. It's a set of 11 domain-based questions that evaluate different types of bias. You apply it based on the study design, so whether you have a cohort study or an RCT. And then the questions are rated by selecting 4 possible answers. So the questions that applied to our particular data sets, the majority applied in some way or another, depending on the case, the study type. For each study, you got a +2 for definitely low risk of bias, +1, -1, or -2. And so essentially, the color coding helps you to see if it was green, meaning more favorable, and red, meaning less favorable with respect to risk of bias. So then this helped us to evaluate the responses for individual studies. So when you were actually looking at what a study reported, we would often look back to see, well, how did it do with confounding? How did it do with exposure characterization? And then how did it also compare to other studies in our evidence base that looked at these similar concepts? So OHAT also recommends that you do what we're calling, they call them a heat map, or risk of bias heat map, where you can look across all of your data and try to see trends. Does this, for example, did this body of evidence do a good job looking at confounding overall? And that helps you to develop conclusions as well. Okay, so integration and synthesis. So this moves into standard 4 and involves using our a priori method for evaluating the evidence and conducting a qualitative synthesis. Again, we did not pursue a meta-analysis or quantitative synthesis in this assessment. So the general flow or content of our syntheses, our narrative syntheses, involved 3 sections. So a general characterization of the literature identified, and we did this for everything, for all of what we found, as well as based on outcomes. And then within an outcome, we characterized the data on an endpoint basis. So everything, we gave a summary based on the individual studies for, so for example, for blood pressure changes or for myocardial infarction. Then we provided plots of wanted to provide plots of findings and our risk of bias figures. Again, this is what we set out in our a priority methods, and then conduct a body of evidence where we integrated both our confidence and developed a conclusion. And then similarly, this was done for the outcome as well. So we wanted to look at the body of evidence, use a weighted evidence approach that also involved our confidence in that underlying evidence, identify strengths, limitations, and uncertainties, as well as data gaps. So this was done primarily following the OHAT framework, which again is based on grade. We selected this when it became available because it is specifically a process for qualitatively evaluating data. So coming up, the OHAT process itself is developing hazard conclusions. And while we weren't really necessarily developing hazard conclusions, it was a good fit for our data set based on the type of anticipated data that we were going to have and the types of conclusions we were going to make. Now, specifically honing in on a piece of that figure that was on the last slide is the section specifically on synthesizing the evidence and rating the confidence. The way that this was done is you start moving from left to right. You start by assigning an initial confidence in your evidence base, essentially based on the study design. So, was there controlled exposure? Did you have exposure prior to the outcome, et cetera? And you make an initial designation. And then you go through a variety of factors that increase or decrease confidence. That might be magnitude of response, risk of bias, et cetera, and come up with a final level of confidence in your body of evidence. Now, how this is actually implemented in our systematic review is shown on this slide. So moving from left to right, we looked for each endpoint. We gave an initial confidence rating, again based on the OHAT handbook. We then looked across the overall risk of bias. So when we looked at that heat map overall for that set of studies, did it seem like there was an overall low risk of bias, a higher risk of bias, and what did that do to our confidence? Also, with respect to that body of evidence for that endpoint, were those studies relatively well designed to evaluate our PICO, or was, for example, caffeine one of hundreds of factors that were evaluated in a study? So what was the author's intent? Now, with respect to magnitude, this is one example of how we had to somewhat tailor some of these parameters to our specific assessment. So magnitude typically, describes the strength of the effect. Was the OR greater than 2? Was it greater than 5? Well, much of our data didn't fit into those typical metrics. But what we were able to do is talk about what was the strength of the effect when there was an effect observed below the comparator. So that's really where we focused on. Since our question was that comparator a safe level? We wanted to know if you saw an effect below, and of course we collected information for above too, but what we're really focused on is when you saw an effect below, what was the magnitude? Confounding played a large role, relatively straightforward in terms of what we were collecting. Again, for each outcome, we had some specific information or specific confounders that we might have been focusing on. So, for example, in reproduction and development, we paid particular attention and weighted the use of pregnancy signal. And then we also looked at consistency. This was a key finding. Were we seeing lots of lack of effects, or were we seeing effects, and how can we integrate those and ultimately designate a final confidence rating? So all of these were then put together and we worked then into developing our conclusion. And this was done using a weight of the evidence approach. Of course, we started with the data themselves. What are the findings saying relative to the comparator? And in particular, when we look at effects both above and below the comparator, what's the context, and vice versa. When we didn't see effects, what was the context? We then developed our confidence in the evidence-based. A lot of discussion went into the consistency and the magnitude of effect, as well as the level of adversity. And this became particularly important when we went from the endpoint to outcome conclusions. So it was more straightforward to develop conclusions for endpoints, but it became more difficult on the outcome level because you were integrating different types of effects, physiological changes that were reversible compared to cardiovascular disease, for example. And then overall, we then used the conclusions from the endpoints and the outcomes to develop an overall conclusion. And the other thing that we set out to do is to report using a common structure. Am I doing that time? Okay, so moving into some of the challenges that we faced and the attempted solutions that we integrated. Many of these came due to the broad nature of our research question. I will reiterate, though, that this is a type of research question that many in toxicology are facing. It's difficult to find very narrow research questions and meet the regulatory needs of toxicology and risk assessment. So these, we think, are not uncommon to the field. Some that I'm going to highlight is the use of healthy, the broad array of outcomes, as well as the definition and integration of adverse. Oops. Okay, so first example I want to give you of the challenge is just thinking about healthy. Much of the literature, and this came about when we were piloting, much of the literature was evaluating caffeine in unhealthy populations or in potentially compromised individuals. And we spent a lot of time talking with our experts, and in particular Dr. Goldberger, about how this may impact our evidence base. And it was ultimately determined that we wanted to only include healthy individuals to streamline and try to get a more accurate look at the effects of caffeine themselves versus caffeine, for example, being used as a therapeutic in a study. So we came up with our definition of healthy, which I mentioned earlier. The impact, however, on our systematic review was such that we did exclude a fair number of studies based on this definition. We did, however, retain a study if it had a healthy control arm. So if the study, for example, the one I'm showing here was in patients with Huntington disease, if we could tell from the title and abstract that it looked like there was a control arm, we would go ahead and look at that to see if we could get any data from those studies. Next, with respect to the outcome, the use of the term adverse. This is something that I think in our field that you could spend weeks talking about. What's adverse and where do you set those cutoffs? And this was a particular challenge for us. It started with trying to determine what we were going to include with respect to outcomes as well as endpoints. And we talked a lot about, should we just use the effects that were reported in NAROT? The reason that we didn't like doing that, though, is that was research published, over a decade ago, and we know that science has advanced quite a bit, and there were likely to be other endpoints that were just not available at that time. So we wanted to be relatively comprehensive and integrate everything that we could to be conservative. However, we also spent a fair amount of time talking about levels of adversity within an effect. I'll use blood pressure as an example. There are changes in blood pressure that are statistically significant versus biologically meaningful. And the more we talked about it, the more we realized it's the context that matters. And it was very difficult to draw a line. And we found ourselves essentially, this could, again, become a research project on its own. And so we, in the end, were very broad and comprehensive in terms of what we brought into the assessment. We did try, as part of our piloting, to integrate the term adverse into our search syntax, but this ended up being too restrictive. As you can imagine, many authors don't actually integrate the term adverse, especially if they're not looking at adversities per se. And let's see, anything else? Oh, and the other way that we tried to think about this, knowing that we were going to have quite an array, is, again, we characterize effects in subgroups based on, for example, physiological, clinical, and we also made notes as to where effects might occur and a progression of effects. Okay, now with caffeine, we felt like we had an added layer of complexity, which is similar to probably many of the agents or things that you think about in nutrition, and that there have been reported benefits, of course, associated with consumption of caffeine. And so it became hard in many cases to draw the line of if you're looking at a benefit or an adverse effect. And what we were seeing when we were piloting is quite a few controlled trials. While they were looking at benefits or therapeutic effects, we did performance enhancing. Did this make somebody cycle faster or run faster? But in it, they have controlled measures of heart rate variability or blood pressure. Similarly, there were quite a few of these types of studies in the behavior category. And so we felt like we were challenged in this way, and that we didn't want to eliminate that rather high quality data. So what we did is we actually included benefit studies, and by that, the author's objective was to evaluate some sort of an enhancement if it included any information on our health outcomes. So we technically have benefit studies in our assessment, but again, we're only focusing on the potential adverse effects in those studies. And something I've spoken a little bit to already is that when we were initiating and piloting and starting to think through this entire process, we knew that we were looking at a very broad set of evidence, and it was difficult to make the case that anything should really be removed. Our decision was really to be comprehensive and inclusive. And this was consistent with what we felt like NAROT had done as well, even though we were taking a systematic approach. Though the one thing that really came true is that we needed to make sure that we were selecting guidance and frameworks to help us facilitate this review that could be flexible and accommodate many different data types, many different dose metrics, many different study types. And that was also emphasis as to why we selected the OHAT handbook and risk of bias tool. I think that I would be remiss if I didn't say that one of the greatest challenges was with data integration. How were we bringing all of this data together? And I think this was even more difficult because we were asking a rather unique question. So we weren't just trying to make a conclusion about potential hazard. Could something happen? We were asking, are there effects at a specific dose? And that is a more risk-based question and what we felt was like a more meaningful question. But it did present challenges when we tried to integrate the data. I wanted to mention that this is something that has been highlighted in a number of regulatory and academic efforts that, with respect to evidence-based toxicology. Even in the NAS review of the EPA IRIS program, this was something that was highlighted as something that you want to have structure to, but it's difficult to have specific guidance for doing that, and that might be because it's on a case-by-case basis. I will also highlight that the EFSA and evidence-based tox collaboration based out of Hopkins is going to have a workshop on this topic specifically, data integration in a systematic review context later this year. So just want to highlight this is still very much an issue on the forefront of developing best practices. We also had challenges when it came to reporting. Thinking back to our original goal, we liked NAROT, we liked the utility of NAROT because it relatively simply gave information on safe levels after characterizing quite a few endpoints. And so now that we had applied the systematic methodology, we were left with a much more lengthy and robust paper, which is inherent to the process and to providing that transparency. Though even getting that into the standard formatting of what you might see in a journal review article was challenging. So the way that we worked with that is that we had not only our protocols registered in Prospero, which assisted with the length of the paper, but also we've utilized supplemental materials of the manuscript, as well as we're putting all of our evidence tables, et cetera, on the data repository from AHRQ. I think with respect to reporting, another challenge that we had was not only in developing, but reporting clear conclusions. So as you saw Dr. Myers present, we have a very structured way of reporting our data and reporting our conclusions. And that was harder to have very simple sound bites, if you will, or simple scientific sound bites, highlights. And so we had to balance that. And we feel like we addressed that by providing consistency in the manuscript, so clear areas of where the conclusions are and how they're structured. And also we provided a very large table in the manuscript which has all of our, essentially the information supporting the conclusions as well as the conclusions themselves. So then I'll just end with a few thoughts on the flow and the timeline of our project. I think this is a question that gets asked quite a bit, and it can fall into the challenges category as well, because that's one of course, the issues that we're facing here is that we want to integrate systematic review, but we also understand that it is a robust process that requires resources and time. So Dr. Myers first presented this project flow diagram. Essentially, this was how the project was implemented, which followed IOM, but went from initiating, and then our next kind of big section or phase was looking at the individual studies, and then our next section was putting it all together, and then our last was reporting it out. I also want to mention that these graphics that say SAB meeting do not represent one instance. These are most often a number of phone calls, numbers of emails, and discussions as well. So the first phase, when we were getting up to speed, this is when we were really starting to get off the ground, was about four months after we had topic formulation a little bit underway. And then it took us about 11 months to get the individual studies, look at them, extract the information, then about 8 months to continue to put all of that together, and that involved not only our analysts within ToxStrategies developing draft syntheses, putting all the materials, making draft figures, but then also discussions with the SAB or the subject expert matters on their particular outcome. So the way it worked is we, before the whole group saw everything, everyone saw their subject matter, their outcome first. And then putting everything together, we went through, of course, multiple iterations of the draft manuscript. The SAB had 2 official rounds as well as a number of, I will call them unofficial comments provided and edits. And then we also, of course, went through the journal peer review process. And with that, I think that ends the method and challenge.